All right, second video, and what we're going to do now is I believe we stopped with the urea broth, so we were here last. Urea broth is also an enzyme test. It starts out this color that's called salmon. I don't know, it looks kind of orangish to me. It says here negative urea test, positive urea test. You're testing for the enzyme urease. Some organisms produce, like Proteus is a bacteria that does this, they produce urease. And what does urease do? Urea is basically from the breakdown of proteins. And any organism that produces urease, urea is called the substrate, urease is the enzyme, and enzymes attach to substrates. And when it does that, urease breaks down urea. And what it breaks it down to is ammonia. Ammonia is basic. And like I said, these media have pH indicators. If there's a change in the pH, either acidic, or in this case, it's actually becoming more alkaline, more basic, you get a color change. So with the urea broth, a negative is this, a positive is this fuchsia hot pink broth. Looks just like that. And what is it positive for? Urease. If you're asked, okay, is this test positive or negative? If it's pink like that, it's positive. What is it positive for? Urease production. Positive urease, hot pink broth. Okay, rabbit plasma. This was the stuff that's kind of watery colored. It looks like this. And this is testing another enzyme test for coagulase. What's the test for? Coagulase production. Coagulase is an enzyme that will clot plasma. It'll cause it to coagulate into a blood clot. Well, into a clot, a solid structure. It's actually watery when it starts out, or a negative is watery. And this is good for distinguishing between Staph aureus and Staph epidermidis. Now I remember what I was going to add. Anyway, so positive coagulase, thick clot like this. You see how these are held at an angle? You can tell that's watery, how it's kind of like laying down like this, whereas this is all solid in the end. Yeah, Staph aureus, stuff found on skin, is always coagulase positive. Staph epidermidis is negative. And you can do this with a lot of organisms, but anyway, it's used to distinguish between the staph a lot of times. So enzyme test, what are you testing for? Coagulase production. A positive is a clot. A negative is clear and watery. Phenylalanine slants. Now this is one of those, it's an enzyme test. You're testing for an enzyme called deaminase. Sometimes it's called phenylalanine deaminase. This particular media has phenylalanine, the amino acid, mixed in. It's kind of a tan color when it starts out. You inoculate it with an organism, you let it incubate, and you're testing to see if that organism produces deaminase. If it does, it will break down the phenylalanine inside the compound. So remember, post-incubation, you have to add this stuff called ferric chloride. It was actually kind of a gold color, yellowish color, really bright yellow. You do that post-incubation. And basically, positive is mossy green agar, negative for phenylalanine deaminase is no color change. These two are negative. That green right there would be a positive. And you can see the yellow that's been added, the ferric chloride. And so what is it positive for? Deaminase. Because it's actually broken down phenylalanine. When you add these reagents, it tests for the compound that's left behind. All right, a few more. These are the SIM plugs. They look like little gel plugs on the ends. They're not auger slants. They test for three different things. The SIM actually stands for sulfur, indole, and motility. Sulfur is hydrogen sulfide, toxic gas. Is the sulfur in the media broken down to produce this toxic gas? It's also testing for this indole production. Let's make that kind of red because it causes a red ring to form. This media has tryptophan, which is an amino acid. If you have an organism that can break down tryptophan, you get a substance called indole. And what you have to do is add a reagent to test to see if indole is present in the media. And it's, yeah, byproduct of the breakdown of amino acid tryptophan. It also tests for motility, and unfortunately in these you really can't see that even here. What you do with this, you use the what are called the inoculation needles. You stab them down the middle all the way to the bottom. If you see any growth away from a stab line, that's motility. In fact, I'll see if I can find a picture and I'll add it to this before I post this for you. See if I can get one that has a motility example. But you're testing for, like I said, you can test for hydrogen sulfide. If you add Covax reagent post-incubation, so like it says there, red ring forming at the top of the plug after addition of Covax reagent. There's an example of it. It's kind of a copper color. It smells really bad. Negative indole will be no red ring forming, and then motility, like I said, is growth away from the stab line. So you have three examples here. That's negative across the board. There's no growth along the stab line. You can't even see a stab line. There's no red ring, no hydrogen sulfide. This one would be positive for indole. You can't tell any motility there. I would say that's negative, and it's negative for hydrogen sulfide. This one is positive for hydrogen sulfide, but you see there's no red ring at the top, so it's negative for indole. And to be honest, you really can't tell if there's motility there or not because it's all blackened like that. So that's called a SIM, sulfur indole motility. Catalase, last example. This is an enzyme test, and you're testing for the presence of catalase. Human cells produce catalase. What catalase does, if you put catalase, if you mix it with hydrogen peroxide, which this is the actual chemical compound, H2O2, it's two hydrogens, two oxygens. Catalase will split hydrogen peroxide into water and oxygen. So water here plus an oxygen. So there's the two. You can see it split off. And there's the O. And anyway, when this happens, it produces a bubbling effect. So what you would do normally is you put a drop on a blank slide like this. You would take your organism on your loop, and you can set the entire loop into that bubble, into the drop of hydrogen peroxide, into the drop of the slide. Okay, yeah, place a drop on the slide anyway, and you see if it bubbles. That's an example. If you put it into a tube, you can do the same thing. It looks like they're putting like almost like a Q-tip stick or something in there. Same thing would happen. Bubbling is a positive, no bubbling is a negative. And what are you testing for? Catalase production. It's differential for catalase production. There we go. Positive means bubbling, negative is no bubbling. Positive, it's splitting the hydrogen peroxide into water and oxygen. It's great for killing anaerobic bacteria in humans because anaerobes are killed by oxygen. So any kind of deep cut, you can pour that in, pour the hydrogen peroxide in. All right, we have reached the end of our review. Now, I'm going to give you a couple of examples. So what I did, I'm going to make a couple of changes before I post that. So what I did here, I'm going to go back. This is week 12. So when you get access to this, here is, okay, the Lab 3 review. Now I got this split up. Here's review documents and videos, which if you're watching this video, you're already there. And then under here is a practice activity. Okay. Well, I'll take that out. I don't have an extra thing in there. So what you're going to do, here is the unknown flow chart. So I'm going to go ahead and click on that. This is the same one that you used for your unknown. So I'm going to go ahead and open this up all the way. Now the first thing you notice, there's a gram-positive side and a gram-negative side. I hope you noticed that the other day. And then I helped you get all your different types of media, or somebody did. Somebody helped you get your media. Make sure you're going in the right direction. First thing you do is a gram stain. You have to see if you're gram positive or negative, and that tells you what side of the chart you're going to start on. So what I did, and I'd like you to do this exactly how I have it posted. Here are 5 unknowns with the numbers. when you click on these, you're going to open up a document like this. So there's the tube you were given. Here's the gram stain. Okay, and then here are all the tests that were run. Assume all these tests are final, all the reagents have been added, and you can see all the color changes, and use these as practice, because part of your exam is going to be you having to interpret a series of tests and trying to figure out what the unknown is. So this is number 10. But anyway, please do that. Do all of them. Go through, look at number 10, pull up your, or have it with you, your flow chart, follow it down, and write down what you have as to what you think the organism is. Okay? There's 5 chances for you to practice. There's 10, 15, there's, oh, I'm sorry, those are the numbers. There's 5 of these. Go through, use the flow chart, and see if you can figure out, without cheating, as far as looking at the answers, what each one of these would be. Down here, answers to practice unknowns, when you click on this folder, it's going to open up and show you the answers. Like, it'll tell you what organism unknown 10 is. I'd like you to do it as practice. Do all 5 of them and then look at the answers. So we're going to do an example first. So here is our first example. This one is number 14. It's not posted. So here's the first thing you would do as a gram stain. We did this in class, or you did this in class with somebody, some instructor. All right, so those are pink cells. If you wanted to get technical, and we're not worried about the cell shape arrangement, those are gram-negative cells. They're either pink or red, and those are rods. It's a bacillus. They look like they're all by themselves. Now, once you figure out, okay, you're a gram-negative, so that means you're going to be on this side of the page. So start looking at the test results. So right here, diagnostic test, look at the results below. Assume all the reagents have been added, like it says here. All results are final. And I made this super big. Here we go. Let's look at the plate first, because that's the first thing on your flow chart. If you look at that, do you see dark purple or green metallic sheen? No. And if you're wondering what's that dark, that's somebody's writing on the back of the plate. But see the lines here? This looks like somebody did a streak for isolation. There's no color change. So that's negative. Okay? You'll want to note that. That's 1. And in your case, if your results, if you had 3 that said it was one thing and 1 innocuous test, you go with the 3. But in this case, so this definitely is a negative for the EMB plate. Indole. Okay, let's look at that one next. That would be the little SIM plug. And you can see right there where the Covax was added. No red ring, no hydrogen sulfide. That's a negative. You see on this chart, it says indole negative, and it says here the hydrogen sulfide can either be plus or minus. So in this case, it's a negative. There was no hydrogen sulfide. Look at the urease next. Urea broth. Remember I said this was salmon colored? It turns hot pink or fuchsia if it's positive. That's a definite negative. So once again, we're kind of looking both here. Urease was negative. Now you're coming down. Now it looks like, so we had negative here, negative here, negative here. So now we're down to these 2 organisms. What do we have? Well, you're going to look at the citrate. Blue slant. Doesn't matter. Any blue change at all is a positive. Eventually this whole thing would turn blue. So that's a positive for the citrate. All right. So the organism is Serratia marcescens. It's actually a red bacteria. Pretty color. That's how you use a flow chart. And like I said, I'd like you to practice. I gave you 5 examples. Do all 5. Write down your answers, or you can just circle them on the flow chart, whatever you want to do. And then, well, write down what you think it is, then look at the answers. Okay, I'm going to close that one out. Let's do another one. Okay, so here's one. Look at this one. Oops. Now, what shape is those? First of all, the color. That is a gram-positive, definitely. And those are causi. In fact, those look like Staphylococcus arrangements. You can see those are circles. Because on our flow chart, you need to note, so, okay, we're gram-positive, so we're going to be on this side, gram-positive bacteria. Now, You need to see if are they rods or are they cocci. And then once again, you follow the flow chart. Now in this case, this one's pretty much already given it to you. It says it's catalase positive. I believe it's still on there. See, it bubbles, so it's definitely positive. And just kind of follow it down. What's the next one? The Msa Auger Plate. Okay, this is red. Remember, a positive for MSA is yellow, bright yellow. And so, yeah, that's definitely a negative. Definitely a negative there. So here it says negative or positive fermentation, and it's talking about that one. So look at the next one, rabbit plasma, coagulase. Can you see that's watery? Because if it was clotted, remember, it'd be stuck in the end. It wouldn't be laid out like this, you know, because if it was a clot, it'd be all collected here, because you store these tubes upside down, or excuse me, the tube, the bottom would be on bottom, top on the top. I could have turned this if I wanted to make it look like that, but that's what you usually do, is you turn it to the side to see if it's clotted. This one did not clot. That's a negative. And then, so you go over here, coagulase was negative. Negative fermentation, of course it bubbled, positive. So now you're down to these two. And you look at the maltose red broth. So here it is, and it's still red. That's a negative. It'd be yellow if it was positive. So it's a negative, so this is Micrococcus roseus. Okay? Yeah, bubbled, cocci. See over here, if it was rods, you'd be on this side. But since it was cocci and gram-positive, you're on this side. We looked at the plate, it was negative. Rabbit plasma, negative. Maltose, negative. Micrococcus roseus. All right, well, I hope this review helps. Good luck on your exam, and thanks for joining me.
And that's the bottom line, because